Our today's topic is structure of bacteria. Bacteria are small, prokaryotic, single-celled organisms found almost everywhere on Earth and are vital to Earth's ecosystem because bacteria are decomposers which break down dead material and recycle it. Prokaryotes have a nucleoid rather than enveloped nucleus. You can see here, this is prokaryotic cell, this is nucleoid without any nuclear membrane. But here in eukaryotic cell, nucleus is surrounded by nuclear envelope, right? Bacterial cell also lacks membrane-bound cytoplasmic organelle. For example, mitochondria, Golgi complex, endoplasmic reticulum, etc. Bacteria have characteristic shapes: spherical, known as cocci, rod-shaped, and spiral bacteria. Bacteria often occur in characteristic aggregates, in pairs, in chains, as tetrads or cluster, etc. Right? Bacteria reproduce by binary fission. Structure of bacteria. Structure of bacteria we will discuss under 4 headings: the nucleoid, surface structures, surface layers, and cytoplasmic structures. Surface structures include flagella, pili, capsules, and surface layers, capsules and loose slime. Cytoplasmic structures include plasma membrane and organelle, right? So first of all, we will discuss nucleoid. Prokaryotic nucleoid. You can see in this figure, this yellow colored structure, nucleoid. It is equivalent to eukaryotic nucleus, right? But simple in structure as compared to eukaryotic nucleus. Eukaryotic nucleus has complex mitotic apparatus and surrounded by nuclear membrane, right? Under light microscope, the nucleoid of bacterial cell can be seen with the help of Feulgen staining. Feulgen staining which stains DNA. So DNA of bacteria is a single, continuous, giant, circular molecule with molecular weight of approximately 3 into 10 raised to the power 9, right? Surface structures. In bacteria, 2 types of surface structures can be recognized. The flagella, this is long thread-like structure flagellum, which are organs of locomotion. Flagella are organs of locomotion. And these small hair-like structures present all over the surface of the bacterial cell, pili, also known as fimbriae, right? Flagella occurs on both gram-positive and gram-negative bacteria, and their presence can be useful in identification and classification of bacteria. For example, flagella are found on many species of bacilli, but rarely found on cocci, right? Pili occur almost exclusively on gram-negative bacteria and are found on only a few gram-positive organisms also. Some bacteria have both flagella and pili. For example, Methanococcus maripulidus. These bacteria have both flagella and pili. First, flagella. Structurally, bacterial flagella are long, 3 to 12 micrometer, and 12 to 30 nm in diameter. Flagella are made up of a class of proteins called flagellin. Flagellin subunits of a flagellum are assembled to form cylindrical surface with a hollow core. A flagellum consists of 3 parts, as you can see in this diagram. A long filament which lies external to cell surface, right? Second is hook structure, which is present at the end of the filament, right? And the third one is basal body, to which hook is anchored and which imparts motion to the flagellum. Basal body traverses the outer wall and membrane structure, as you can see in this diagram. It consists of a rod This is rod and one or two pairs of discs. These are discs. Movement of the basal body is driven by proton motive force rather than by ATP directly. What are ATPs? ATPs are energy-rich molecules, right? Ability of bacteria to swim by means of propeller-like action of flagella provides them with mechanical means to perform chemotaxis. Now, what is chemotaxis? Chemotaxis is a movement of an organism in response to a chemical stimulus, right? Number and distribution of flagella on bacterial surface are characteristic for a given species, hence are useful in identification and classification of bacteria. In bacteria, 4 type of flagella are present. So flagella type number 1 monotrichous, second lophotrichous, amphitrichous and peritrichous. In Vibrio, monotrichous type of flagella are present. In monotrichous, single flagellum is present at one pole of the cell. Lophotrichous, here more than 2 flagella are present at one pole of the cell. Third type, amphitrichous. In Spirillum species, when more than 2 flagella are present at both the poles of the cell. And the last one, peritrichous, for example, in the case of Escherichia coli, where flagella are distributed all over the surface of the bacterial cell, right? Next, pili. Term pili and fimbriae are usually used interchangeably to describe thin hair-like structures present on the surface of many gram-negative bacteria. Pili are made up of group of proteins called pillin. Pili are more rigid in appearance than flagella, right? These are pili present on the surface of the bacteria. Pili can come in 2 types, short and abundant, commonly present pili, right? And the second type, long pili, but less in number. Number ranges from 1 to 6. These pili called sex pili, right? Pili in many bacteria confer adhesive properties to the bacterial cells, means enabling bacteria to adhere to various epithelial surface, means to red blood cells, to surfaces of yeast and fungal cells, etc. Sex pili. Sex pili can be distinguished by their ability to bind male-specific bacteriophage, means sex pilus acts as a specific receptor for these bacteriophages, right? Sex pili also attach male to female bacteria during conjugation and facilitate DNA uptake and transfer through conjugation. As you can see in this diagram, this is donor cell as it possesses sex pilus and F plasmid. Now, what is this F plasmid? F plasmid is a structure which is characterized by the presence of a factor known as fertility factor or sex factor. It helps transfer genetic material from one bacteria to another through conjugation. So, first, this sex pilus helps in attachment, means attachment of male to female bacteria during conjugation. So first attachment, then transfer of genetic material, right? So when genetic material get transferred from donor to recipient also becomes donor. You can see here old donor and new donor. Next, surface layers. Principal surface layers are capsules and loose slime. Cell wall of gram-positive bacteria and in complex cell envelope of gram-negative bacteria. Then plasma membranes and mesosomal membrane vesicles, which arise from invagination of plasma membrane. How the mesosomal membrane vesicles are formed, we will discuss later. In bacteria, cell wall forms a rigid structure of uniform thickness around the cell. And This structure is responsible for characteristic shape of the cell, right? Inside the cell wall is plasma membrane. This is usually closely opposed to the wall layer. Capsules or loose slime. Some bacteria form capsules. Now you can see here this is a hard green colored structure, capsule, which constitute the outermost layer of the bacterial cell and surround it with relatively thick wall of viscous gel. Capsule may be up to 10 micrometer thick. Some bacteria lack well-defined capsule but have loose, as you can see here in this diagram, loose amorphous slime layers external to the cell wall or cell envelope, right? Gram-positive and gram-negative bacteria differ drastically in organization of the structure outside the plasma membrane but below the capsule. Gram-positive and gram-negative terms we use for bacteria because gram-positive, have distinctive purple appearance after gram staining, but gram-negative are pale reddish in color after gram staining because they lose crystal violet color, right? So gram positive and gram negative, they differ in the organization of the structure outside the plasma membrane. In gram negative organism, these structures constitute the cell envelope, whereas in gram positive organism, they are called cell wall, right? Now, intracellular components. First, plasma or cytoplasmic membranes. Plasma membranes of animal cells contain four major phospholipids: phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and sphingomyelin, which together account for more than half of the lipid in membrane, right? These phospholipids are asymmetrically distributed between the two halves of the membrane bilayer, as you can see here in this diagram, it is shown. The outer leaflet of the membrane consists mainly of phosphatidylcholine and sphingomyelin, right? Whereas phosphatidylethanolamine and phosphatidylserine are the predominant phospholipids on the inner leaflet of the membrane, right? Next, mesosomes. In gram-positive bacteria, vesicular or tubular vesicular membrane structures called mesosomes are formed. How? By an invagination of plasma membrane. You can see here, this is cell wall, this is cell membrane. It invaginates and form mesosome. This is mesosome. These mesosomes, means these type of structures, are much more prominent in gram positive than in gram negative bacteria, right? Now, other intracellular components. In addition to the nucleoid and cytoplasm, the intracellular compartment of the bacterial cell is densely packed with ribosomes. Ribosomes which are present in bacteria are of 70S type. These ribonucleoprotein particles, which have a diameter of about 18 nm, are not arranged on membranous rough endoplasmic reticulum as they are in the case of eukaryotic cells, right? Other granular inclusions randomly distributed in cytoplasm of various bacterial species include metabolic reserve particles such as poly-beta hydroxy butyrate (PHB), polysaccharides, and glycogen-like granules, and polymetaphosphate or metachromatic granules. This is all about structure of bacteria. Thank you.